So we've worked our way down to the most inferior part of the brainstem, the medulla oblongata. This structure, along with the pons, forms the ventral wall of the fourth ventricle. Now we're going to pause here in this area and talk about this fourth ventricle and its proximity to the structures of the pons and the medulla for just a moment. And I'm going to tie that together with several pieces and parts for you to give you something, a real take-home message there. It contains choroid plexus. This is not new. So we've seen this structure made of ependymal cells and capillaries secreting cerebrospinal fluid in all the ventricles, the two laterals and the third. The pyramids we've also seen. So we're going to look at a section, a coronal or cross-section, depends on how you want to say that. I always refer to them as these particular sections as coronal. That's the way we did them in rat brains, but that's not important. But we're going to look at the pyramids and talk about them again today. Remember, these are the axons of the corticospinal tracts that we've seen. And this area, of course, is where, in these pyramids here, is where the fibers cross, where 85% of the somatic motor fibers cross over and then run down the lateral funiculus of the spinal cord to the particular spinal segment and then form a synapse in the lower motor neuron, and off it goes to the muscle. All right, so this is a section of medulla that I'm going to use to talk about a couple of times today. There's several interesting pieces and parts here. You can note the nuclei of a couple of cranial nerves. I'm going to point that out here in just a minute. I don't intend for you to be able to locate them in a section like this, but I want you to be well aware of the cranial nerves associated with this part of the brain. I'm also going to show you this nucleus today. Talk about it for just a minute, the solitary nucleus, an important connecting piece for the autonomic nervous system. You'll see both the vestibular and cochlear nuclei today. These are all very important strategic stations for information in the medulla oblongata. The presentation that I showed you online mentions the reticular formation, and you'll remember, if you watch carefully, these reticular formation are dark gray matter areas that extend throughout the brainstem, and then they make far-flung connections throughout the cerebral cortex. And you can see them clearly here. You can see why these are called a large cell group and this is a small cell group. You can see that in this picture, obviously. And the raphe nucleus is another very interesting structure of the reticular formation. There's a picture on the presentation here that talks about sleep. And it's got some connections in there. There's some mention of the raphe nucleus there. And I'm going to wait on that. It takes me a little while to go through that. But in physiology, I'm going to connect some sleep information for you that your book doesn't. And then I normally in the spring try to take kids over to the sleep center at Cabrini and let you get a sense about how they do that. We can do EEGs in the lab, but I want you to see that, and then we'll talk a lot more about the SCN and the structures in the diencephalon, the structures in the brainstem that are interconnected for describing this phenomenon we call sleep. So I'm not going to do that this semester. You'll see the image, but I'm not going to talk about the details of it. This is just to point out to you this reticular formation. So you're seeing things here. You're looking vestibular and cochlear. You're seeing nuclei of cranial nerves. You're seeing reticular formation. You're seeing down here the pyramids, the medial lemniscus tract, which we've already talked about, this major sensory pathway from the fasciculus canetus and gracilis. We can see it passing through the medulla here. This inferior olivary nucleus, I think comes up on the next slide, where I'll tell you that this is a major relay point for sensory information, for somatic sensory information. These inferior olivary nuclei play important roles in delivering somatic sensory information to the cerebral cortex. So that was not intended for you to write all that down. I just wanted you to see these pieces and parts and get some sense about where these things are in the medulla. And now I want to focus in on, after I mention these, I want to focus in on the key moments, the key points that you're going to take away with you. Okay, so of the nuclei of the medulla, the inferior olive, big one. Nobody mentions the medulla without mentioning the inferior olivary nuclei. These are gray matter areas that relay sensory information. I cannot describe for you how difficult it is for me just to say that. I want to go down into the cord and track this, but there's no time. And it's not really my job. I've given you a lot of motor and sensory pathways already, enough to get you a baseline. These are famous sensory, somatic sensory for controlling muscles in your body, these inferior olivary nuclei of the medulla. We also see, as we've done in every other brain section that has cranial nerves attached to it, I've pointed these out to you. Where does 1 attach? Where does 2 attach? Where does 3 attach? Where does 4, 5, 6? Where does 7, 8, 9? Where does 10, 11, 12? Where do these nerves attach to the brain? So 10, the vagus, 11, the accessory, and 12, the hypoglossal, are all connected to the medulla. The vestibular nuclear complex is here. These are synapses that help regulate and maintain equilibrium. I do have a picture in here to show you that pathway. I'm not going to do that. That pathway does include the superior olive, which is very close to this one. But I'm just going to show it to you. I'm not going to make you memorize the pathway for the balance information. I'll just let you see it. So this is a very important balance structure in the medulla. There are cranial nerves here. There's major somatic sensory information being transferred here. And then there are ascending sensory tract nuclei, including the cuneatus and the gracilis. So this is the connecting piece for what we did in the dorsal funiculus, so the spinal cord. So if I asked you about the fasciculus, cuneatus, and gracilis in the spinal cord, you have a sense about where that is, sacrocervical, in the dorsal funiculus. These are the areas in the medulla where that information is traveling through, that fine touch information on its way to the thalamus. So these are what I would call the big four here, or major pieces of the medulla. Now, so I show that picture again. So remember, I know vestibular. This is the hearing piece here. I didn't mention that, but these maintaining cranial nerve 8 information, right, is passing through to the medulla. The inferior olive here, major somatic sensory information, the pyramids, the medial lemniscus, also major sensory information. All these structures, you don't need to memorize where they are, just have a sense about what they do and that they're in the medulla. All right, now this is the picture I said I'm not going to beat you up with. You can see here, let me point out to you what you recognize. You recognize this structure right here, the inferior colliculus. That's a midbrain structure we talked about. That guy right there is the superior colliculus. When I mentioned inferior colliculus to you, I said the inferior colliculus is a sensory relay station, specifically for auditory information. Hearing information is passing through the inferior colliculus of the midbrain. So this picture shows you that, and I mentioned it to you before. Look, you recognize that word? The lateral lemniscus. Now I haven't shown you the lateral lemniscus, but you saw the medial lemniscus, a major somatic sensory band traveling up to the thalamus. The superior olive, which I mentioned the inferior olive on the last one. Again, I'm not going to force you to learn this pathway. I'm just showing you some things that you have seen, some things that you haven't. This labels the dorsal cochlear and ventral cochlear nuclei here of the medulla, which you saw in the last picture. And this shows you where the nuclei are here in the medulla, which patterns with the other one. And this shows you the hearing and balance structures. So again, I know it's dangerous to say this to students, but don't worry about this. Although, if I had said medial geniculate, or if I had said superior temporal gyres for auditory, or if I had said inferior colliculus here, or if I had said lemniscal tracts, then you should have known something about that already. So this pathway is another one that's interesting, but I'm not going to go through the details of it for you. Just to show it to you here, the medulla is receiving these types of sensory information. And I typed them out for you, so you need to memorize that. Okay. Now, I do want to emphasize these parts because when you're talking about the medulla and physiology, these are the places where we're really going to focus in on control things. So for physiology next semester, we're going to talk a lot about the control of the cardiovascular system and a lot about the control of the respiratory system. And in those conversations, we're going to include structures of the medulla oblongata. So I like to introduce the structural pieces here in the fall semester. Okay, now this picture's not in your book, and you first look at this, you go, ugh. Okay, but it's actually a very simple image to help students get a structural framework about the connections in the medulla that are so important for the cardiovascular and respiratory system. All right? So let's identify first the nuclei of the medulla. NTS stands for the nucleus of the tractus solitarius. All right, so let me go back and show you this. I said I was going to show it to you. So you can see in this picture right here, the solitary nucleus labeled right here in the medulla. This is the place where we're talking about right here, the nucleus of the tractus solitarius. Big fancy name, the solitary nucleus of the medulla. This is a major location. This NTS is a major location for really important autonomic information, stuff that is going to impact the cardiovascular system. Okay, so I want to show you that in just a second. Also notice the rostroventrolateral and the caudal-ventrolateral medulla. Now I'm not going to split those up. You will not see RVLM or CVLM on your test. It's just to say that these are medullary nuclei. And you can see that I'm going to use these guys right here to talk about cardiovascular respiratory things. Okay, so the first part of this is to show you the nuclei of the medulla. You don't need to memorize where they are, but you need to be aware that there are medullary nuclei that are important for cardiovascular and respiratory function. These are them. Okay. Now out here on the left-hand side of the picture, you can see this is the common carotid artery and the external and internal carotid artery. And this spot right here, where the common carotid becomes the external and internal, is a spot called the carotid sinus. Now, that has come up in this class already. So let's come back to it in just a second. Here's another very important anatomical location for connections in the medulla, the aortic arch. Now, this first one, the carotid sinus, labels cranial nerve 9, and it has information arrow pointing this way to the medulla. Cranial nerve 9 into the nucleus of the tractus solitarius. This one has cranial nerve 10 into this area. So evidently, cranial nerve 9 and cranial nerve 10 are connected to nuclei of the medulla. The information that cranial nerve 9 and 10 are delivering to the medullary nuclei are critical. They are critical pieces of information for the cardiovascular and respiratory system. Y'all see the connection now? I've mentioned it before, but it's really important. It's worthy of pointing out to students over and over again. Cranial nerve 9 and cranial nerve 10 carry sensory information about heart rate and about blood gases that are critical for processing things for what we're going to do with our blood pressure and our heart rate and our breathing rates. This information traveling along these two nerves. All right, now you can also see here, coming out of the medulla, an arrow going this way. And this information appears to be going to the heart or somewhere around the heart. It doesn't show specifically where it's going. But you've learned the anatomy of the parasympathetic motor outputs. And so you should be able to describe for me this connection of the vagus to the heart. you should already have some idea about that anatomical connection. You should say something like this: preganglionic parasympathetic information is associated with cranial nerves 3, 7, 9, and 10. And this information in cranial nerve 10, leaving the vagal nuclei of the medulla, traveling along the vagus nerve, is headed to ganglion cells that are located at targets in the thoracic and abdominal body cavity. One of those targets is the heart. And so this preganglionic vagal output here is going to synapse with ganglion cells at the heart. And then the postganglionic fibers are going to influence the heart, specifically heart rate. The vagus slows the heart rate down. So medullary nuclei, vagal nuclei of the medulla, send this information to slow down the heart. So there is both sensory and motor. There are both sensory and motor pieces of information on the same nerve, autonomic sensory and autonomic motor. And they're connected directly to the medulla. Now you can also see here, there's another piece, you can see here that there are connections within the medulla. There are connections within the medulla that then connect with neurons in the spinal cord. So medullary nuclei connect with neurons located in the spinal cord that are part of the autonomic nervous system. These neurons are connecting, these neurons from the medullary nuclei are connecting in the spinal cord to neuronal cell bodies in the lateral horns of the spinal cord. And the good anatomy student hears that and they immediately go, that's sympathetic information. So you can see the medulla has not only an impact on the heart, impact on receiving both cardiovascular and respiratory information, but you can also see here that the medullary nuclei are connected to the sympathetic nervous system. So this picture is intended to teach you review on parasympathetic and the influence of the vagus on the cardiovascular system. And it's also intended to teach you, reteach you again, the impact of the sympathetic nervous system, the connections of the sympathetic nervous system. We call the sympathetic nervous system the thoracolumbar division. That's what we called it. And we said the preganglionic cell bodies of the sympathetics are found in the lateral horns of T1 to L2. And then they send their presynaptic or preganglionic connections out to ganglion cells, where they make synapses. And then from those ganglion cells, postganglionic projections then go to the targets. Now in this case, you can see that the targets are arteries and veins. Now as I look through this simplified diagram, I saw an artery up here, but I did not see any information going this way. And then I saw an artery here, and I did not see information going this way. And then I saw aortic arch here, and then I know from my anatomy before that this connection is not to the arch. This motor connection is to the rate determining structures. This is a connection to the SA node. And so at this point, you're supposed to realize, you're supposed to have an aha moment here where you say to yourself, now wait a minute, are you suggesting to us that the influence, the connectivity between the medulla and the cardiovascular system through the sympathetics is the only way to get autonomic motor information to blood vessels? That is exactly what I'm suggesting to you. So when we talk about the cardiac inhibitory center and the vasomotor centers in the spring, I'm going to say this again, we're not going to learn the connections, but I'm going to talk about the specific physiology on how this happens, right? These neurons here in the spinal cord, influenced from the medulla, control the sizes of the blood vessels in our body. And that is a purely sympathetic phenomenon. It's very unusual to see structures that don't have both parasympathetic and sympathetic input. This is an unusual phenomenon. You're supposed to recognize when you see this, arteries and veins, you're supposed to go, now wait a minute, why don't I have nuclei from 3, 7, 9, or 10 going to these blood vessels? You're supposed to recognize that because the smooth muscle of the walls of arteries and veins is exclusively controlled by sympathetics. Now, there are a couple of exceptions to that, but if you learn it that way, you got most of it. Now, the questions undergraduate students have when they first see this, when it first hits them, is, now, wait a minute, I thought blood vessels have to get bigger and smaller. There's a lot to be learned about how we distribute blood flow in our body. So when we do this in physiology, we're going to beat this horse to death. How in the world are you going to do that if you only have one system targeting these things? So sometimes I need more blood in my digestive system. Sometimes I need more blood in my brain. Sometimes I need more blood to my heart. And you don't have enough blood to do all of that. So how do you distribute blood flow? The sympathetic nervous system is responsible for that. And the only way to understand how that's possible is to talk about the physiology of the receptors found in the smooth muscle cells. So that will launch our discussion of alpha and beta adrenergic receptors and how they work, how the chemicals do what they do to regulate the distribution of blood. In anatomy, you're supposed to get this. It is sympathetics in connectivity with the medulla, right? We'll start there. The vasomotor system communicates with the lateral horn neurons of the sympathetic nervous system, and they, in turn, deliver the information to the blood vessels for constriction or dilation. The story of how you make some big and how you make some small is the story of the physiology. This shows you the connectivity. So I love this picture. That's why you're getting it. It's a great image to have in your head about the regulation that the medulla is involved in. All right, now, let's go one step further. I promised this just a moment ago. So over here on this picture, this is a picture I use in physiology. Well, let's start with this one. Okay, so over here, you can see something that I mentioned on the presentation to you. The pons we didn't talk about much. We connected cranial nerves there, but we didn't talk about much of many of the nuclei. Most of the structures in the pons are fibers of passage, things passing through. But I mentioned to you, there are some pontine nuclei that influence breathing. Okay, but they don't drive it. They influence it, they don't drive it. The drivers of breathing, as you can see here, are medullary nuclei. Now, whenever I teach students the connection of C1 and C2, the DENS and the odontoid process, I always mention this. I say to the students, if in fact you are to push the DENS, if it busts the transverse ligament, holding it in place, and it penetrates into the anterior part of the brainstem, it can kill you because it can damage respiratory centers. These are the respiratory centers here, showing you these respiratory centers, and you can see the connection here. In fact, you could do better with this, I'm guessing. You could take my respiratory center information here from the medulla, you could as a student, and you could connect that information out of the brain into the spinal cord on your way to the diaphragm. You would go to the lower motor neurons of segments C3, C4, and C5. Coming off of those segments, you would form the phrenic nerve. And the phrenic nerve would then come from the spinal cord and pass down to the diaphragm to make the diaphragm contract. If I pointed over here to the external intercostal muscles, you would say, okay, I can do that too. I can take my motor information out of the medulla, I can take that information down the spinal cord, and I can choose the segmental thoracic spinal nerves and go to the muscles of the muscles of the chest in a segmental fashion, T1, T2, T3, T4, T5, and I can make those muscles contract. Now you also would be aware from my illustration about the dens that the group of nuclei of the medulla that are mainly responsible for this are on the front side. Y'all see that? This is the anterior side. There's the cerebellum, this is the front side. So those nuclei are right there. These axons passing, you see they're right in that critical anterior portion of the brainstem. And you know where they are? You know where these structures are whenever you get to the spinal cord as well. These axons here passing through the spinal cord are in the lateral funiculus on the dorsal side, cervical to sacral. So these would be up in the cervical regions, over in the central portions of the spinal cord. Okay, so I know you don't like doing that, okay? But in my opinion, you can't learn all the anatomy, so find the things that are important and do them over and over again so that you never forget them. And this is a really important one. All right, now, the reason this picture is here is not for that. The reason this picture is here is to show you something that I'm going to talk about a lot in physiology. So on this image, you heard me say cranial nerve 9 and 10 deliver sensory information to medullary nuclei. You heard that from the carotid sinus and the aortic arch. So obviously, there's information about something going to the medulla. And I told you this is information about heart rate, blood pressure, respiratory gases. So that's a very important connection to the medulla. This picture indicates information coming from the medulla going the other way. So you thought on this one, you thought to yourself, there is information out of the medulla going back to the heart, and that does have an influence on the heart. That's parasympathetic information, and it slows the heart down. That's autonomic. On this picture, you thought to yourself, this is somatic motor information coming out of the medulla, influencing breathing. So I have information coming from the aortic arch and the carotid sinus coming to the medulla, and I have information leaving the medulla going to the heart to influence it, and I have information going from the medulla to the spinal cord to influence the blood vessels for blood pressure. So I have respiratory and cardiovascular information on a two-way highway with the medulla. Now this is the last thing I'll point out to you because I do want to spend some time on it. This picture shows you, this image over here is supposed to indicate to you this area of the brain over here. Really these deep structures over here of the brain. The fourth ventricle should extend a little bit further here. These are medullary structures here. These are neurons of the medulla oblongata. And you can see that there are dendritic endings of these neurons that actually sample the cerebrospinal fluid of the fourth ventricle. Now, this picture shows you a very famous respiratory reaction that will become second nature to you. I gotta explain acid-base balance. This is the equation. So you can see all these different ions here, but the point today is to show you that there are medullary structures sampling the fluid of the fourth ventricle, specifically for the purposes of respiration. These guys actually bind hydrogen, which is a byproduct of elevations in CO2. And when they do, they will drive breathing. So I want to finish with this. The structures of the medulla oblongata drive breathing from both the sensory and the motor perspective. So you are receiving sensory information from cranial nerve 9 and 10. You're delivering motor information via the phrenic nerve. You are receiving information at the medulla itself about blood gases. And then using that information, that's what this picture is intended to teach, using that information to then deliver somatic motor information for breathing. This is, compared to the influence of cranial nerve 9 and 10, this one far supersedes it. Every textbook calls this connection, this medullary connection here, calls it the central control center for breathing. The central control center, these central chemoreceptors are responsible for driving the breathing here. And they do it by sampling the CSF for blood gases. Okay, so I like to finish with that, so you got a real sense about the importance of this, right? This is what I wanted to get. Did you get this hammered into your head? Cardiovascular and respiratory centers, sensory and motor information associated with the medulla oblongata. All right, the last brain structure is the cerebellum to talk about, and we won't spend a lot of time on it. I have a few things to say about it. It is located dorsal to the pons. It protrudes under the occipital lobes. It makes up a big chunk of brain, right? 83% of the brain is cerebrum, 11% is cerebellum. How much is that? 94. So everything else we've talked about, from the mesencephalon through the brainstem, makes up about 6% of the mass. So the cerebellum is huge mass. We know that it provides precise timing for appropriate patterns of skeletal muscle contraction. Now, I gave you a little of that. I gave you a taste of that. I want you to tag here for me, because it is the standard. Tag the sensory path for the corticopontine circular connectors that I did for you, starting with the dentate, the Purkinje to the dentate, passing through the superior cerebellar peduncles. I want this pathway to go in your head right here for providing these precise time information. You should also think about the cerebellum in terms of what it's going to do. If I have a pathway to influence skeletal muscle contraction, which you do, I taught it to you already. If I have that pathway, then I should also have some way of receiving sensory information in the cerebellum. And you already have that piece of information as well. So I made a circuit for you. I connected the pontine nuclei back into the cerebellum so that you can see you can influence this in a circuit from the cerebellum. You can influence muscle contraction. But I also told you, when I showed you the spinal cord, that there is sensory information on the lateral edges, proprioceptor information from the muscles in the joints, passing along the lateral edges of the cerebellum, along the lateral edges of the spinal cord up to the cerebellum. So this means, anatomically, this means more to you. It's supposed to already mean more to you here for the cerebellum. All right, and this activity occurs subconsciously. That's an interesting statement. We'll not belabor the point there. Okay, the anatomy. There are two bilaterally symmetrical hemispheres connected medially by the vermis. The last time you saw the vermis here, we were talking about the dural septa. And I told you that the falx cerebelli travels along this midsection of the cerebellum. The tentorium cerebelli, remember, goes into the transverse fissure, and the falx cerebri goes into the longitudinal fissure. The falx cerebelli travels along this vermis, the middle part of the cerebellum. It has folia. I think about a Rolodex here, transversely oriented gyri. Each hemisphere has 3 lobes. Now, I have a couple of pictures here where I'm going to show you what these lobes do. But I realize that you have learned a lot of connectivity already. So pay close attention to the things that I focus on, because I'm not going to make you learn all of the details of these cerebellar connections. I want you to come away from me with some big picture idea about what this part of your brain does. And y'all, here's the real interesting part here. We don't really know. Nobody really understands exactly what the cerebellum is doing. We know it's involved with cognition. We know it's involved with motor control, both sensory and motor pathways. We've got the track tracing. We've seen what damage to Purkinje neurons can do. We know that there are diseases related to the cerebellum for motor control and balance issues and the way people move. We know that. But there's some other things about the cerebellum that are a long way off from being figured out. For example, the connectivity in the cerebellum is beyond, just in this piece of the brain, is beyond the scope of anything ever imagined by man with electrical connections. I'm going to give you some numbers on that here in just a minute so you can see that. And I have a couple pictures on these nodes to show you how they're, and these pictures are not intended to force you to learn brand new pathways. They're intended to say, look, I told you this before. Look at what the cerebellum is involved with. It is important. We think about it as a subconscious brain. Nobody thinks about the cerebellum in functional things, but it is important. Okay, there's gray matter area and cortex. We call that white matter projections in the cerebellum. It looks like a bush. When you cut it in a sagittal section, you look at it from the sides, lateral view, you look at that, it looks like a bush there. And the anatomists, original anatomists, were all seminary guys, and they were looking for ways to interject their faith into their naming. And so this is one of those places. They named this little tree the Tree of Life. Isn't that interesting? So all subsequent generations after the original naming would have to think about the Tree of Life, the Arbor Vitae, when they labeled the white matter of the cerebellum. This is a tree-like pattern in the cerebellum when you look at it from a lateral view. All right, we know that it receives impulses involved in muscle contraction. You should be able to do better on this. I taught you, again, go back and review that corticopontine loop so you can have that. Proprioceptor and visual signals inform the cerebellum of the body's condition. You do not have this pathway from me. I did not teach you how the visual information gets to the cerebellum, but I did talk about this one, the proprioceptor information, located in the lateral edges of the spinal cord. Okay, now we think this is what it does. It calculates the best way to perform a movement. Man, there's a lot of hiding going on here in terms of how the cerebellum does that. But you have pathways in the central nervous system and connections from the peripheral nervous system to the cerebellum that you can use to explain this. So when I see a statement like this about the cerebellum and what it's doing to calculate movement and involved in movement patterns, I think, yeah, I got you. Here's what I got. As the sensory information comes from my Golgi tendon organs all over my body, and from the spindles, the muscle spindles that are located in the muscles, it travels along these spinal nerves. And when it gets to the spinal cord, it passes through the dorsal root into the cord. And as it passes into the dorsal horn, those axons are distributed out into the lateral edges of the spinal cord. So they're going to the cerebellum, this proprioceptor information passes out to the lateral edges, and then the axons, up they go along the edges of the spinal cord to the cerebellum. Now when they get to the cerebellum, they initiate this pathway that the cerebellum uses to influence muscle contraction. And that pathway should be thought of as beginning with the Purkinje nuclei, projecting through the dentate nuclei, then through the superior cerebellar peduncle into the midbrain, a large somatic relay station called the red nucleus. Am I ringing some bells? The red nucleus then projects its information up into the cerebral cortex, where it can directly influence the neurons that are then going to deliver somatic motor information to the muscles. That information in the cortex can also be delivered back to the cerebellum again by way of the corticopontine tracts. So there's a looping piece of information there in the cerebellum. So I have a way up, I have a way down, and I have a way of doing sophisticated something, calculating things in the cerebellum. You have the neuroanatomy in place in order to think about these observations. All right? So here's the vermis, and this is shows you the lobes. You don't need to memorize pictures here. This shows you the arbor vitae, and here's its position in relationship to the fourth ventricle. Just images that you guys are very, very familiar with now. And then this picture shows you what I meant by don't learn all the pathways, but show them some things about these nodes, what they're capable of doing. Just give them something to hang their hat on. Okay, so I've marked these things with dark red lines. Do you see them? Things to hang your hats on. And can you guys see why the pieces and parts here have dark red lines on them? Why would your teacher, you know me well now, why would he put the dark red lines on the lines that are here? Let me just say the words. Ventral lateral nucleus of the thalamus. Corticospinal tract. Vestibulospinal tract. Why would I put dark red lines on these pieces? Because I'm not trying to add more to your life here. I'm trying to repeat things that I have said before. So in the original picture of the outline, you learned the cerebellum and its connections. And then we talked about the cerebellum and its connections as we have gone through this thing. I mentioned it when we got to the midbrain. We did that whole circuit thing, and I keep bringing it up over and over again. So this shows you that the different areas of the cerebellum are connected to these very famous pieces that you have already learned. Y'all, there is nothing new here. You should recognize these named structures. Now, we didn't talk about this one, the lateral forebrain bundle. We didn't do that one. But we mentioned this one in passing. We mentioned this one in detail, and that one in detail, and that one in detail, and that one today. So there's nothing here for you to memorize in addition to what you've already learned. I just want to show it to you. The cerebellum is connected, and you can do that. You guys can do that now for the cerebellar connections. Now, as we finish this, I'd like to try to impress you with the numbers. So in a histology section of the cerebellum, whenever you do this for Dr. Du Bois, you will learn that the cerebellum is organized into 3 layers that are very distinct from one another and very easy to locate histologically. I took the histo out, yeah. This was to redo the structure that I've already done twice today. But this one right here is the way I introduced the histo. 3 distinct layers. There is a molecular layer, the most outer layer of the cerebellar cortex. There's a middle layer called the Purkinje cell layer, and all the students go, Purkinje cell layer? Yeah. I know, I got you, right? This is where my pathway started with the Purkinje cell layer. That's what he taught us before for the influence of the cerebellum on muscle contraction. Okay, and then there's a granular layer. Now, what I didn't tell you before, and I don't ask numbers questions like this on tests, but this is intended to say, you gotta be kidding me. The Purkinje cell layer is a layer of the cerebellum that's easily identified. And the Purkinje neurons, you can see them here, are some of the biggest neuron cell bodies in the brain. You can clearly see them, and a lot is known about them. The Purkinje neurons appear to be major inhibiting connectors because they make loads of this neurotransmitter called GABA. They're inhibitory neurons, and they're really easy. They're beautiful in sections, histosections. You can see them. Each Purkinje neuron has 1,000 or more contacts. Okay, so a neuron is a small thing, okay? And when I draw these things in physiology, I normally draw one cell connected to another cell. And all the pathway stuff that you've learned so far, right? I go from here to there, to there, to there. 1, 2, 3, 4 connectors, right? First order, second order, third order neurons. The cerebellum's doing something else sophisticated. because each of the cells of the Purkinje layer has more than 1,000 synapses on it. How in the world are you going to process that kind of information? Some of them are excitatory, some of them are inhibitory. Okay, but it's worse than that. The granular layer of the cerebellum, the third layer down, has 50 billion neurons in it, which comprises up to 75% of all of the cells in the central nervous system. Now, it would seem, it seems obvious to me that if you're going to talk about the importance of a structure, you shouldn't spend time talking about 25% of it, right? But that's what we have done in neuroanatomy. We have spent all of our time talking about 25% of the total cell volume, because 75% of it is in the cerebellum. And we do that for good reasons, I think, because we're trying to explain how things are connected and how they work. But this is flabbergasting to me. The number of connections and the number of cells here is so far advanced, and so who knows what comes out of the cerebellum in the future. There's a lot not known here. All right. I have two things left to say about the nervous system, and then on Friday we're going to start cardiovascular. Let me think about that for just a minute. Let me hold off on that decision. Tomorrow in the lab, we're going to start the blood vessel dissection because it takes a few weeks and we only have a few weeks left. So you've done your brains. If you haven't labeled those structures, you haven't watched those videos that are available online, do that before we get started tomorrow in lab. I need you here. See that dirty look? I need you here at 12:45 so I can tell you how to get started. Because your last major dissection for this course is the cardiovascular system. So all the time spent dissecting has really been for muscles at this point. First half of anatomy, muscles. Second half, blood vessels. So there's a few other things to do, right? But this is where you will spend the bulk of your time. We're going to get it started tomorrow. And I need to give you some instructions. 12:45. Get the cats out. Go ahead and get here early. Get your cat out. And at 12:45, I'm going to say a few things about the blood vessel dissection. And then you're going to be ahead of me a little bit in lab. But I'll catch up. That's it.